Following blood collection, the quick detection and identification of diseases is carried out with the help of different reagents in laboratories. With the immunoassay method, very small quantities of substances can be detected from the blood. The production of these reagent kits is one of the most important tasks of this business unit. The applied RIA method is based on the idea of a Nobel Prize winner. Preparation of coated tubes is a one-week process. Different solutions must be administered every day with an appropriate machine. A special material has to be applied to the inner side of the plastic tubes, which is able to bind biotin-containing compounds. This special material is streptavidin. Since these tubes are used in diagnostic kits in healthcare, their quality control is essential. The coated tubes must be protected against moisture and dust. Therefore, they are hermetically sealed. In vitro diagnostic tests are based on tracers that use isotope technique. Biomolecules attached to radioactive isotopes detecting hormones, proteins and peptides with high sensitivity are important in studying disease. To produce high-quality tracers, sufficiently pure labeled material is needed. During the labeling process, the radioactive isotope is chemically bound to the molecule. As many impurities are present or formed in the reaction mixture that adversely affects the quality of the labeled material, a purification step is needed during the manufacturing process. The tracers are purified by chromatographic methods. The inactive liquid phase component of the kits is dispensed in the clean room. Here, such conditions have to be maintained that protect both the staff and the products from possible microbiological contamination. This can be achieved by installing the appropriate working environment and by complying with personal hygiene standards and rules laid down in the operational standards. In the dressing room, operators change their clothes, shoes, and put on face masks and hairnets. The materials and equipment are transferred to the operation area through airlock following a preliminary disinfection. Dispensing and stoppering of the vials takes place here. Lyophilization, also called freeze-drying, is a dehydration process that ensures the long-term conservation of pharmaceutical and food products. Lyophilization is the most advanced and the gentlest drying method, preserving the biological activity of sensitive materials. Lyophilized products can be quickly and easily reconstituted. The business unit lyophilizes numerous materials. In this way, the stability of sensitive components can be guaranteed until they are used. Recently, a high degree of automation took place in clinical laboratories. The aim was to reduce labor costs, to increase the number of measurements performed during a given period of time, and to improve the repeatability and reproducibility of the measurements. The business unit develops and manufactures kits to be processed automatically. After placing the reagents, samples, and test tubes into the measuring device, the automat pipettes, according to the selected protocols, forwards the test tubes to the incubator, and they go through a washing station before finally arriving at the gamma counter. The results are received after an automatic evaluation. The final stage of production is the packaging of the kits. Hundreds of pieces are made from more than 80 types of kits each day. The components of the kits are stored in cold storage rooms at different temperatures until their packaging. The most important part of packaging is a two-step checking. After checking, the kits are covered with a shrink foil and put into a cold storage room for temporary storage until their distribution. Customers are served with the kit containing the appropriate components, instructions for use and control report.